This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome. Today, we explore a profound biological mystery. Why does all life on Earth share just one single origin? We'll journey back 4 billion years to meet our common ancestor, Luca, and investigate the failed experiments that may have flickered in Earth's early laboratories. From the closed door of our oxygen-rich atmosphere to the haunting possibility of a hidden shadow biosphere, we'll examine the chemistry, competition, and staggering mathematical odds that defined our beginning. Join us as we uncover why the spark of life became a one-time-only event. Chapter 1. One Code, One Ancestor. When we look at the staggering diversity of life on Earth today, from the microscopic bacteria thriving in a drop of pond water to the blue whales navigating the deep oceans, it is easy to assume that such variety must have sprung from many different beginnings. However, the deeper we look into the molecular machinery that powers every living thing, the more we find a profound and singular unity. Every organism we have ever studied, without exception, operates on the same basic chemical operating system. This realization has led to one of the most significant conclusions in modern biology. All known life on this planet is part of a single, massive family tree that traces back to one specific point of origin. This point is represented by the last universal common ancestor, often referred to by the acronym LUCA. The most compelling evidence for this single origin is the universality of the genetic code. To understand why this is so significant, we have to look at how life stores and uses information. Life uses a 4-letter alphabet of nucleotides in DNA, known as adenine, thymine, cytosine, and guanine, often abbreviated as A, T, C, and G. These letters are arranged into 3-letter words called codons. Each of these codons provides the instruction to add a specific amino acid to a growing protein chain. For example, the sequence of the 3 nucleotides ATG always corresponds to the amino acid methionine. Theoretically, there are countless ways a genetic code could be structured. There are dozens of amino acids found in nature, yet life on Earth consistently relies on a specific set of 20. Furthermore, the correspondence between a specific codon and a specific amino acid appears to be essentially arbitrary. There is no fundamental chemical law that dictates ATG must mean methionine rather than another amino acid. If life had emerged multiple times independently, we would expect to see a variety of genetic languages, perhaps some organisms using different sets of nucleotides or different codes for translating those nucleotides into proteins. Instead, we see the same code used by a blade of grass, a mushroom, and a human being, with only minor variations in a few rare lineages. This shared language points to a single moment of success in the distant past. It suggests that while many different self-replicating molecules might have existed on the early Earth, one specific system became so efficient and dominant that it established the standard for everything that followed. This ancestor lived roughly 4 billion years ago, and although it was likely a simple single-celled organism, it was already incredibly complex. It possessed hundreds of genes encoded in the same DNA code we use today. This implies that by the time Luca appeared, the transition from simple chemistry to complex biology was already well advanced. The complexity of this shared ancestor is further evidenced by the cellular machinery required to translate the genetic code. Every living cell uses a suite of specialized tools to turn DNA instructions into functional proteins. This includes messenger RNA, which carries the code from the DNA, transfer RNA, which brings the correct amino acids to the assembly site, and ribosomes, the massive molecular factories where proteins are built. The fact that the core of the ribosome is made of RNA rather than protein is a molecular fossil that hints at an even earlier stage of life. But the ribosome itself is universal. Whether it is in a bacterium or a human cell, the ribosome performs the same catalytic work in the same way. This creates what scientists call a chicken and egg problem. A cell copies its DNA using an enzyme called DNA polymerase. But that enzyme is itself produced by translating the DNA polymerase gene stored in the DNA. Neither the enzyme nor the genetic code can be produced or utilized without the other. The existence of this tightly interlinked system in every branch of life is a powerful indicator that we all inherited it from a single, highly refined ancestral line. Beyond the genetic code, the basic metabolic pathways used to extract and store energy are also remarkably consistent across the tree of life. All organisms use a molecule called adenosine triphosphate, or ATP, as their primary energy currency. The process of creating this energy often involves chemiosmosis, where a cell pumps protons across a membrane to create a gradient, then uses the flow of those protons back through a molecular turbine to synthesize ATP. One of the most ancient metabolic routes, the Wood-Ljungdahl pathway, allows certain microbes to turn carbon dioxide and hydrogen into energy and organic matter. This pathway is found in both bacteria and archaea, the two primary branches of life, suggesting it was present in their common ancestor. Genomic research has identified roughly 355 genes that can be traced back to LUCA, and the functions of these genes tell us a great deal about its life. It appears this ancestor was anaerobic, meaning it lived without oxygen, and likely inhabited a high-temperature environment rich in minerals, such as a deep-sea hydrothermal vent. Another striking piece of evidence for a single origin is the phenomenon of homochirality. Many organic molecules are chiral, meaning they exist in two forms that are mirror images of each other, much like a left and right hand. In non-biological chemical reactions, these two forms are usually produced in equal amounts. However, life is incredibly selective. All proteins in all living organisms are made exclusively of left-handed amino acids, while the sugars in DNA and RNA are exclusively right-handed. If life had started multiple times, it is statistically probable that some lineages would have used the opposite-handedness. The fact that all life shares the same chirality is a strong indication that we are all descendants of a single chemical event that happened to favor one orientation over the other. This unity is also visible in the hierarchical pattern of relatedness that we see when we compare the genetic sequences of different species. By looking at genes that are highly conserved, meaning they change very slowly over millions of years, scientists can construct a tree of life. This tree shows that humans are more closely related to chimpanzees than to gorillas, and more closely related to mammals than to reptiles. But if you go back far enough, we share a common ancestor with fungi, plants, and even the bacteria found in yogurt. This is not a metaphor, but a documented biological fact. Every living thing you see is connected by an uninterrupted chain of reproductive events, going back billions of years to that first successful lineage. It is also important to consider the first-in, best-dressed principle. Once the first life form emerged and began to proliferate, it fundamentally changed the environment of the Earth. These early organisms would have quickly consumed the primordial soup of organic molecules that had built up over millions of years. Any new proto-life attempting to emerge through abiogenesis at a later date would have found itself in a world already populated by efficient, highly evolved competitors. In a very literal sense, the first successful life form likely closed the door behind it, making it nearly impossible for a second, independent origin of life to gain a foothold. This ancestor established the biochemical rules of the game, and those rules have governed the evolution of every creature that has ever lived since. While we can reconstruct the characteristics of this ancestor through genomics and biochemistry, the path that led from simple molecules to this complex system remains a mystery. We know that the early Earth provided the necessary ingredients, water, minerals, and energy sources like lightning and geothermal heat. And we know that simple organic compounds like amino acids can form naturally under these conditions. We have even found these building blocks on meteorites and in interstellar space. In fact, polycyclic aromatic hydrocarbons, PAHs, are the most common polyatomic molecules in the observable universe and were likely a major constituent of Earth's primordial sea. This suggests that the raw materials for life are common in the universe. Yet, the transition from these simple bricks to a self-sustaining, self-replicating cell capable of Darwinian evolution is a gap in our knowledge that spans hundreds of millions of years. This gap represents the period between the formation of the Earth and the appearance of the first uncontested fossils. These are the stromatolites, layered structures created by colonies of cyanobacteria that date back approximately 3.5 billion years. They represent a time when the first experiments in life had already transitioned from a single ancestor into a global, landscape-altering phenomenon. The era preceding these ancient fossils remains the great mystery of our origins, a time when the first experiments in life were taking place. This singular history suggests that while the ingredients may be common, the specific path to a successful, enduring biological system might be a rare and complex journey. Chapter 2: The Hadean Laboratories and the Ghosts of Failed Life. While the molecular evidence presented in the previous chapter confirms that all modern life descends from a single common ancestor, this biological unity is likely the result of a brutal process of elimination rather than a solitary miracle. The story of life's beginning is often framed as a lone spark in a dark world, but the laws of probability and the sheer scale of the early Earth suggest a much more crowded and chaotic picture. Before the lineage of our ancestor established its global monopoly, the planet was likely a vast network of independent chemical experiments. This hidden history suggests that life did not just start once. It flickered into existence many times, in many different forms, only to be snuffed out by a planet that was still finding its own stability. To understand why multiple origins are not just possible, but statistically probable, we must look at the nature of the Hadean Earth. In 1983, researchers David Raup and James Valentine used mathematical models to explore the diversification and extinction of the first molecular systems. Their findings suggested that if life had only originated once, the risk of total extinction would have been incredibly high. In a world of shifting tectonic plates, massive volcanic eruptions, and a constant rain of asteroids, a single lineage of proto-life would have been extremely fragile. For life to have a realistic chance of surviving into the present day, it is far more probable that there were multiple independent origins, perhaps as many as 10 or more. According to these stochastic models, even if multiple forms of life emerged, the odds are that over hundreds of millions of years, all but one would eventually go extinct due to random chance or competition. This leaves us with the single origin tree we see today, but it masks a much more diverse and competitive past. The environment of the Hadean eon, spanning from the Earth's formation about 4.5 billion years ago until roughly 4 billion years ago, was a planetary-scale meat grinder. David Deamer has described this era as one of frustrated life. Imagine a shallow sea or a hydrothermal vent where chemical reactions have finally reached a level of complexity that we might call alive. Just as this new lineage begins to replicate and spread, the Earth is struck by a massive asteroid during the late heavy bombardment. These impacts were not mere craters. The largest had the power to volatilize the entire ocean, turning the seas into a thick blanket of scorching steam and sterilizing the surface. Any life that had managed to take hold would have been vaporized instantly. This cycle of emergence and destruction could have repeated for hundreds of millions of years, with life as a frequent visitor to the early Earth, only to be reset to zero time and again by cosmic interference. During this window of planetary chaos, different laboratories across the planet were likely running their own independent experiments in parallel. One such laboratory could have been the deep-sea alkaline vents. In the microscopic, honeycomb-like pores of these mineral structures, different chemical systems could have evolved in total isolation. One vent might have produced a system based on simple RNA-like molecules, while another vent, hundreds of miles away, fostered a metabolism-first system that relied on iron-sulfur minerals to catalyze reactions. However, the surface and the vents were not the only options. The deep hot biosphere theory, championed by Thomas Gold, suggests that life may have originated miles below the surface within the Earth's crust. In this scenario, the deep rocks provided a stable, sheltered environment far removed from the asteroid impacts and ultraviolet radiation of the surface. This isolation would have allowed for the development of lineages that were fundamentally different from anything emerging in the oceans above. These subterranean pioneers would have subsisted on the chemical energy of the rocks themselves, potentially forming a vast hidden network of life that existed entirely independently of the surface biosphere. Other laboratories focused on different chemical pathways entirely. One compelling model involves a specific chemical path using hydrogen cyanide, hydrogen sulfide, and ultraviolet light. This combination has been shown in laboratory settings to produce over 50 different nucleic acids simultaneously, providing a rich starter kit for a genetic system that might look very different from our own. While we rely on a specific set of nucleotides, these cyanide-based experiments might have produced lineages with entirely different genetic backbones. In contrast to these high-energy environments, some of life's first starts may have occurred in cold laboratories. A remarkable experiment involving ammonia and cyanide left in a freezer for 25 years demonstrated that even at sub-zero temperatures, complex chemistry can progress. This experiment produced 7 different amino acids and 11 types of nucleobases. This suggests that icy moons, icebergs, or frozen primordial ponds could have served as slow-motion crucibles for life, fostering lineages that thrived in the cold and were perhaps destroyed when the planet eventually warmed. Complementing these frozen crucibles were environments where heat and evaporation drove the chemistry forward. Sidney Fox demonstrated that by heating and drying mixtures of amino acids, they could be induced to spontaneously form long, complex chains called proteinoids or polypeptides. These experiments suggest that the simple cycle of rain falling on hot volcanic rock and then evaporating could have generated a vast abundance of protein-like structures, providing a massive library of chemical tools for the first protocells to utilize. These thermal laboratories would have been scattered across the volcanic archipelagos of the Hadean, each one churning out a slightly different variety of these polypeptides. Even the medium of replication may have been different in these early trials. The clay theory suggests that the first replicators were not organic molecules at all, but silicate crystals in common clay. These crystals can store and transfer information from mother to daughter crystals through their physical structure, acting as non-organic replicators. In this view, organic molecules like RNA were originally just hitchhikers that learned to use the clay crystals as a scaffold before eventually taking over the process of replication themselves. This implies that there could have been many different mineral-based lineages that never made the jump to organic chemistry, remaining as a form of proto-life that has long since been ground back into the Earth's crust. We may even see the ghosts of these multiple origins in the world of viruses. Some virologists suggest that viruses are not merely escaped genetic material from cellular life, but may represent independent lineages that never evolved into full cells. The fundamental split between RNA viruses and DNA viruses could point to separate abiogenesis events, one that mastered RNA and another that mastered DNA, which only later began to interact with the cellular lineages that survived.
In this light, viruses are not just parasites, but the last surviving remnants of alternative experiments in self-replication that occurred alongside the birth of our own ancestor.
If these multiple origins did occur, the question remains why they all seem to have vanished. One possibility is that our single origin isn't actually single at all. Instead of one lineage destroying the others, the different starts might have merged through a process of rampant horizontal gene transfer. In the very early stages of life, before the development of sophisticated cell walls, genetic material was likely much more fluid. This was an era of a communal pool of genetic inventions. One lineage might have invented a particularly good way to build proteins, while another perfected a way to store energy. Through the constant swapping of genes and the merging of simple vesicles, these different inventions could have been combined into a single, highly refined system. Our common ancestor, Luca, might actually be a biological greatest hits album, a composite of all the successful experiments happening across the early Earth. This communal origin theory helps explain the profound differences between the two primary domains of life, bacteria and archaea. While they share the same genetic code, they have fundamentally different cellular hardware, such as the fats in their membranes and the way they replicate their DNA. It is possible that bacteria and archaea represent two independent starts that emerged from the same communal environment. They may have shared the operating system of the genetic code through gene swapping, but went their separate ways when it came to building their physical structures. We see a powerful analogy for this extinct diversity in the later history of life with the Ediacaran biota. These strange frond-like and quilted organisms dominated the oceans about 600 million years ago, yet they look almost nothing like the animals that appeared later during the Cambrian explosion. Most of these Ediacaran lineages went extinct without leaving any modern descendants. They were a failed or replaced experiment in multicellularity. If such a massive turnover could happen with large visible organisms, it is almost certain that similar turnovers happened at the microscopic level during the Hadean. There may have been entire kingdoms of RNA-based life or alternative biochemistry microbes that dominated the planet for millions of years, only to be wiped out by a change in ocean chemistry or the arrival of a more aggressive competitor. The possibility of multiple origins even extends beyond our own planet. Some scientists propose that life might have originated on Mars and been transported to Earth via meteorites. During the early history of the solar system, Mars cooled faster and likely became habitable while the Earth was still a molten hellscape. If a life seed started on Mars and traveled to Earth, it might have arrived to find that a native Earthling lineage had already started in a hydrothermal vent. These two alien lineages would have then met in the early Earth's oceans, kicking off the first interplanetary competition. Our current biology could be the result of a Martian immigrant outcompeting a native Earthling, or a hybrid of the two. This concept of life arriving from elsewhere even led Francis Crick and Leslie Orgel to propose the directed panspermia hypothesis. They suggested that the arrival of life on Earth might not have been a cosmic accident involving meteorites, but rather a deliberate act of seeding by an advanced extraterrestrial civilization. While this remains a highly speculative theory, it underscores the profound mystery of how life began. For some of the most prominent minds in biology, the jump from chemistry to life seemed so improbable that they looked to the stars for an intentional author. Whether life arrived by meteorite or by design, it would have entered a world already teeming with its own indigenous chemical experiments. The transition from this chaotic era of multiple starts to the unified world we know today was likely driven by the increasing complexity of life itself. As metabolic pathways became more tightly linked, the cost of changing the basic rules became too high. Once a lineage reached a certain level of sophistication, it could no longer easily swap parts with unrelated systems. The communal phase ended and the individual phase began, effectively closing the door to new abiogenesis events.
The very success of our ancestral line created a world where the conditions that allowed it to emerge were being consumed or altered, leading to a winner-take-all scenario that would define the future of the planet. One of the most persistent questions in the study of our origins is why we do not see new life forms popping into existence today. If the laws of chemistry and physics are universal, and if the early Earth was able to bridge the gap between inanimate matter and living cells in a relatively short geological window, it seems logical to wonder if that same bridge is still being crossed in some hidden corner of our modern world. However, the scientific consensus suggests that the very success of the first life forms, combined with a shift from a reducing to an oxidizing atmosphere, has effectively sealed the borders of the biological kingdom. The transition from non-life to life, known as abiogenesis, is not merely a matter of having the right ingredients. It requires a sterile and highly specific environment that has long since vanished from the face of the Earth. The realization that life does not spontaneously emerge in our modern environment was famously cemented in 1859 by Louis Pasteur. Through his elegant swan neck flask experiment, Pasteur demonstrated that even a nutrient-rich broth would remain sterile if protected from airborne contaminants. By showing that life only arises from existing life, a principle known as biogenesis, he definitively disproved the ancient theory of spontaneous generation, which suggested that organisms like maggots or mice could simply materialize from decaying matter. While Pasteur's work was a triumph for medicine and hygiene, it also highlighted a profound mystery for the study of origins. If life only comes from life in the modern world, then the conditions of the early Earth must have been fundamentally, almost unimaginably different.
To understand why a second genesis is so unlikely, we must look at the most obvious obstacle, the overwhelming presence of life itself. In the modern world, there is no such thing as a truly sterile natural environment. From the freezing depths of the Antarctic ice to the scalding plumes of deep-sea hydrothermal vents, life has colonized every conceivable niche. This creates a massive problem for any nascent proto-life attempting to emerge. In the prebiotic world, simple organic molecules like amino acids, sugars, and nucleotides could accumulate in primordial soups or concentrate on the surfaces of minerals without interference. They had millions of years to interact, bond, and slowly build toward complexity. Today, however, those same organic molecules are seen as a high-energy food source by the quintillions of bacteria and archaea that inhabit our planet. Charles Darwin himself recognized this problem in the late 19th century. He speculated that if a protein compound were chemically formed in some warm little pond today, it would be instantly devoured or absorbed by existing creatures. This would not have been the case before living creatures were formed. This observation has been reinforced by modern microbiology, which describes the Earth as a septic environment. Every drop of ocean water, every gram of soil, and even the air we breathe is teeming with microscopic organisms that have spent nearly 4 billion years perfecting the art of scavenging. These organisms possess highly specialized enzymes designed to break down organic matter with incredible efficiency. Any random chemical assembly that starts to look like a self-replicating molecule would be an easy meal for a modern bacterium. The competition is not just about being eaten. It is about the insurmountable gap in capability. Modern microbes are the result of billions of years of refinement. They have developed sophisticated defenses, efficient metabolic pathways, and the ability to rapidly adapt to changing conditions. A newly emerging protocell, barely functioning and not yet capable of defending its internal environment, would face an impossible struggle. One vivid analogy used by researchers is that attempting to start life today would be like putting a baby in a ring with an all-time heavyweight champion who has held the title for 2 billion years. The established lineages of life have already occupied all the environmental niches and consumed the available chemical resources. The free-for-all chemical buffet of the early Earth is gone, replaced by a world where every scrap of energy is accounted for by an existing biological system. This biological saturation is complemented by the concept of microbial infallibility. This is the idea that there is no naturally occurring organic compound that some microbe, somewhere, cannot learn to eat. The diversity of modern microbial life is so vast that it is difficult to imagine any new start remaining undetected or unconsumed for long. We only know how to culture about 1% of all known microorganisms in the lab, which means the other 99% are out there in the wild, occupying every possible environmental niche and exploiting every possible chemical resource. Any new chemical system attempting to organize itself would be broken down into its constituent parts before it could ever reach the stage of a functioning cell. Beyond the biological competition, there is a more fundamental chemical barrier, the presence of oxygen. For most of us, oxygen is synonymous with life, but in the context of abiogenesis, it is a potent poison. For the first 2 billion years of Earth's history, the atmosphere was reducing, meaning it lacked free oxygen and was instead rich in gases like methane, ammonia, and hydrogen. This environment was conducive to the formation of complex organic molecules because there was no oxygen to tear them apart. In a reducing atmosphere, the chemical reactions required to build the precursors of life are energetically favorable. This all changed roughly 2.4 billion years ago during an event known as the Great Oxygenation Event. Photosynthetic cyanobacteria began producing oxygen as a byproduct of their metabolism. Initially, this oxygen was absorbed by minerals, causing the Earth to literally rust on a planetary scale. Once the minerals were saturated, oxygen began to build up in the atmosphere. Because oxygen is a highly reactive and corrosive element, it actively attacks and degrades carbon-based molecules. It acts as an electron thief, disrupting the delicate chemical bonds that hold together long chains of peptides and nucleic acids. The modern oxidizing atmosphere is the antithesis of the environment needed for life to form from scratch. When we see fat go rancid or iron rust, we are witnessing the destructive power of oxygen. For modern life to survive in this environment, it has had to evolve a suite of complex proteins and enzymes specifically designed to repair the damage caused by oxygen. We have molecular machinery dedicated to shielding our DNA and cell membranes from oxidative stress. A nascent life form emerging today through abiogenesis would lack these billion-year-old defense mechanisms. It would be chemically unstable and would likely disintegrate before it could even begin to replicate. The very atmosphere that sustains us today would be lethal to the fragile, unguided chemistry of a second genesis. This shift in atmospheric chemistry also changed the way energy flows through the environment. The Miller-Urey experiment, which successfully produced amino acids from inorganic precursors, relied on a highly reducing mixture of gases. When similar experiments are conducted using the modern atmospheric composition, the yield of organic molecules is drastically diminished or non-existent. The addition of oxygen creates a chemical wall that prevents the spontaneous assembly of the building blocks of life. While some organic molecules still form in space or in isolated pockets like hydrothermal vents, they are no longer able to accumulate in the concentrations or varieties required to spark a new biological lineage. Furthermore, the very tools that nature used to build the first life forms have been removed from the workshop. In the weakly reducing atmosphere of the Hadean, certain minerals acted as essential catalysts for abiotic amino acid synthesis. Specifically, iron and carbonate minerals were abundant and accessible, providing the reactive surfaces necessary for complex chemistry. Today, those essential catalysts are largely locked away. The Great Oxygenation Event caused these minerals to precipitate out of the oceans, forming the massive banded iron formations that we now mine for ore. By sequestering these minerals in the geological record, the Earth has effectively hidden the catalysts required for a second genesis. The chemistry of the oceans has also undergone a radical transformation. Research into the Hadean Earth suggests that the early oceans were slightly acidic, with a pH of approximately 5.5. This acidity played a crucial role in the solubility of minerals and the energetics of early metabolic reactions. Modern oceans, by contrast, are slightly alkaline, with a pH around 8.1. This shift in pH represents a significant chemical barrier. The specific proton gradients that many scientists believe were the spark for the first metabolisms, particularly in alkaline hydrothermal vents, rely on a specific difference between the vent fluid and the surrounding seawater. As the global ocean chemistry changed, the battery that could have powered a second abiogenesis was effectively drained. The saturation of the Earth's surface by life has also altered the physical conditions that might have aided abiogenesis. In the prebiotic world, wet-dry cycles in tidal pools or on volcanic rocks were likely essential for the polymerization of molecules. As water evaporated, it would concentrate monomers, forcing them to bond into longer chains. When the pool refilled, these new polymers could interact and potentially find their way into lipid vesicles. Today, these cycles are constantly interrupted by biological activity. Microbes form biofilms on surfaces, trapping nutrients and altering the local chemistry. They change the pH of their surroundings, consume the concentrated minerals, and physically occupy the spaces where chemical reactions might have once taken place. Life has essentially colonized the laboratory, making it impossible to run the same experiments that nature performed 4 billion years ago. Even the deep-sea hydrothermal vents, which many scientists consider the most likely cradle of life, are no longer the pristine chemical environments they once were. These vents are now home to complex ecosystems of tube worms, crabs, and specialized bacteria that have evolved to thrive on the chemical energy pouring out of the Earth's crust. These organisms are incredibly efficient at capturing the hydrogen, methane, and minerals that would have been the raw materials for a second abiogenesis. The white smokers and black smokers are no longer quiet chemical reactors. They are crowded cities where every available molecule is being fought over by highly evolved residents. Another factor is the depletion of the interstellar pantry. During the Hadean Eon, the Earth was subject to a heavy bombardment of meteorites and comets, which may have delivered millions of tons of organic prebiotic elements per year. While cosmic dust still falls to Earth today, the rate is significantly lower, and the massive influx of ready-made building blocks has largely ceased. The early Earth was a unique crossroads of intense energy, high-velocity chemical deliveries from space, and a protective, oxygen-free atmosphere. That specific combination of factors was a one-time-only deal in our planet's history. This biological and chemical saturation has created a closed-door effect. Once a self-sustaining chemical system capable of Darwinian evolution is established, it fundamentally changes the rules for everything that follows. It consumes the precursors, alters the atmosphere, and fills the available space. This is why we see a single tree of life rather than a forest of independent origins. Our ancestor was not necessarily the best possible form of life, but it was the first to become efficient enough to monopolize the planet. By the time any other starts could have gained momentum, the Earth had already been transformed into a world that was hospitable for the descendants of the first winner, but hostile to any new contenders. The transition from the prebiotic era to the biological era was a one-way street. The emergence of life was not a single event, but a process of increasing complexity that eventually reached a tipping point. Once that point was crossed, the Earth was no longer a gigantic, productive, outdoor chemical laboratory for abiogenesis. Instead, it became a biological world where the laws of ecology and natural selection took over from the laws of pure chemistry. The conditions that allowed the first cells to form, the reducing atmosphere, the acidic oceans, the available mineral catalysts, and the absence of predators, were destroyed by the very life they helped create. While the modern Earth is a vibrant and diverse place, its current state is a testament to how life has locked down the planet's resources. The Great Oxygenation Event was perhaps the most significant turning point, creating a world where carbon-based molecules are under constant chemical attack. This, combined with the scavenging power of modern bacteria, ensures that any new proto-life would be snuffed out in its infancy. The story of life on Earth is a story of a single, massive success that was so complete it made its own repetition impossible. This environmental and biological lockdown is the primary reason why, despite the vast stretches of time that have passed since the first cells appeared, we remain a planet with only one known biological history. If the door to a second Genesis is truly locked by the very presence of our own biology, we are forced to look beyond the obvious. We must ask if the first successful lineage truly eradicated all its rivals, or if some alternative experiments managed to retreat into the shadows, surviving in the fringes where our modern probes have yet to reach. Chapter 4. The Hidden Forest. Searching for the Shadow Biosphere. Despite the universal biochemical language discussed in Chapter 1, a fascinating and controversial possibility remains. What if we are not alone on our own planet? This is the concept of a shadow biosphere, a hypothetical realm of life-supporting systems consisting of microorganisms with unique or unusual biochemical structures that may have once existed or might even still exist alongside our own. The idea is that while our particular lineage became the dominant force on Earth, other independent origin events could have produced different forms of life that simply operate outside the range of our current scientific detection. These weird life forms would be the descendants of a second or third Genesis, living in the cracks and corners of the world, effectively invisible to us because our tools are designed only to see ourselves. Cleland points to desert varnish, a thin, dark, manganese-rich coating found on rocks in arid regions, as a potential signature of this hidden world. Despite decades of study, the origin of desert varnish remains a mystery. It forms much faster than can be explained by pure chemistry, yet no known standard microorganism has been shown to produce it in the lab. Cleland suggests that desert varnish might be the product of a shadow biosphere, microbes that generate energy by oxidizing minerals in a way that is fundamentally different from our own metabolism. These organisms might thrive in extreme environments where our form of life struggles, such as the high radiation environment of the upper atmosphere, or the isolated pores of the deep continental crust, serving as a modern refuge for lineages that never joined the global biological collective. The history of science provides a powerful precedent for this kind of oversight, demonstrating that major branches of life can hide in plain sight for centuries. Until the 1970s, the massive domain of the archaea was entirely unknown, mistaken for common bacteria until molecular sequencing revealed their unique chemistry. More recently, the discovery of giant viruses, starting with the mimivirus, further illustrated our biological blind spots. Mimivirus was so large and possessed such a complex genome that it was initially dismissed as a bacterium. It was only when researchers looked past their expectations of what a virus should be that they realized they were looking at a form of life that challenged the traditional boundaries of virology. If an entire class of giant viruses could remain undetected in a common cooling tower, a shadow biosphere with a radical biochemistry is well within the realm of possibility. This biological dark matter represents a vast unknown. Scientists have noted that we only know how to culture about 1% of the microorganisms we observe in the environment. The remaining 99% are known only by their genetic signatures, and even then, we find massive amounts of genetic material in the soil and sea that does not match any known branch on the tree of life. While most of this is likely undiscovered diversity within our own lineage, the possibility remains that some of it belongs to a completely different biological tree. While the prebiotic diversity discussed in earlier chapters provided a vast array of building blocks, our lineage's reliance on a narrow subset suggests that other experiments could have utilized the remaining chemical palette to form a parallel biology.
A shadow biosphere might be composed of organisms that use a non-standard set of amino acids or alternative nitrogenous bases, giving their proteins and genetic structures different properties than our own.
The phenomenon of molecular handedness offers another clear dividing line for a potential second genesis, as a mirror image life form would be separated from us by the chirality wall established in Chapter 1. Beyond chirality, researchers are intrigued by the possibility of life that uses alternative elements in its core architecture. While standard life is built on a foundation of 6 elements, some hypotheses suggest that non-standard life could have gained a foothold using elements like arsenic in place of phosphorus. It has been proposed that standard phosphate-based life eventually gained a specific evolutionary advantage over these non-standard competitors, leading to the current monopoly we see today. This suggests a period of intense competition in the early Earth's history, where the superior stability and energy transfer capabilities of phosphorus allowed our ancestors to outpace and marginalize alternative biochemistries, pushing them into the extreme niches where they might still linger. This concept of isolation helps resolve the tension between the single ancestor model and the multiple origins theory, representing lineages that did not participate in the communal gene-swapping pool discussed in Chapter 2. The existence of a shadow biosphere would prove that abiogenesis was not a once-in-a-universe fluke, but a robust and perhaps even inevitable consequence of planetary chemistry. It would suggest that the landscape of potential biological systems is vast, and that our particular form of life is just one small island in a much larger sea of possibilities. Mapping this chemical landscape is a major goal of modern astrobiology, as it helps us understand what to look for on other worlds. If Earth can harbor multiple independent biospheres, then the chances of finding life on Mars, Europa, or Titan increase dramatically. The search for these other Earthlings requires a new generation of tools. Scientists are now developing agnostic life detection techniques that do not rely on specific genetic sequences or metabolic pathways. These include instruments that look for unusual concentrations of complex molecules, or for evidence of chemical disequilibrium that cannot be explained by non-biological processes. We are also looking closer at the biological dark matter in our own backyard, using advanced sequencing techniques to look for orphaned genes that do not seem to fit into the known tree of life. Every time we explore a new extreme environment, a subglacial lake in Antarctica or a hypersaline pool in the Mediterranean, we are looking for that one organism that doesn't work the same as everyone else. The discovery of such an organism would force us to redraw the tree of life, or perhaps to accept that there is not one tree, but a forest. It would show us that the unity of life we see today is not a fundamental law of nature, but the result of a long competition that our ancestor happened to win. This uncertainty about what we might be missing on Earth naturally leads back to the fundamental question of the odds. If life is so robust that it might have started multiple times here, we must reconcile that with the staggering mathematical challenges of creating even the simplest cell. Finding a shadow biosphere would suggest that the bridge from chemistry to biology is much easier to cross than our current models indicate, shifting the entire debate between chemical inevitability and a once-in-a-universe fluke. Chapter 5. The Odds of Existence. At the heart of the mystery of our origins lies a fierce mathematical conflict. On one side is the argument that the transition from chemistry to biology is so complex that it represents a once-in-a-universe fluke, a statistical impossibility that we only witness because we are the lucky winners of a cosmic lottery. On the other side is the belief that life is a matter of basic chemistry, an inevitable consequence of the laws of physics operating over vast scales of time and matter. This debate is not merely philosophical. It is built on rigorous calculations of probability, information theory, and Bayesian statistics that attempt to quantify the unreasonable likelihood of our existence. To understand the skeptical view, we must look at the sheer scale of the assembly problem. Skeptics often invoke Borel's law, a statistical principle suggesting that events with a sufficiently low probability, often cited as 1 in 10 to the power of 50, simply do not occur within the lifespan of the universe. When we apply the laws of probability to the assembly of a living system, the numbers quickly challenge this threshold. Consider a single protein of average length, perhaps consisting of only 50 amino acids. Since life uses 20 different types of amino acids, the number of possible combinations for that one small protein is 20 to the power of 50, or roughly 10 to the power of 65. This single molecule already exceeds the limit of Borel's law.
If we assume that even a minimal cell requires hundreds of such proteins to function, the odds of these components coming together by chance in a single moment are essentially zero. One famous estimate compared this process to the probability of a tornado blowing through a junkyard and successfully assembling a functional Boeing 747. The calculation can be taken even further by looking at the total number of attempts the universe has had to create life. Based on current scientific estimates for the age and size of the observable universe, there are roughly 10 to the power of 80 atoms. If we assume 10 to the power of 12 atomic interactions occur every second, and that the universe has existed for nearly 14 billion years, we arrive at a total of 10 to the power of 110 possible attempts for a correct chemical combination to form. When compared to the 1 in 10 to the power of 5,000 chance of randomly placing amino acids into the correct positions for the proteins needed for the simplest life, the math suggests that life should not exist anywhere in the universe.
However, many researchers argue that these calculations are fundamentally flawed because they assume life must appear all at once, fully formed. Instead of a single giant leap, the modern scientific view describes a process of many small, cumulative steps. In this framework, the math changes dramatically. A helpful analogy is the rolling of 500 dice. If your goal is to get all sixes in a single throw, the odds are astronomically low. But if you are allowed to keep every 6 you roll and only re-roll the remaining dice, you will reach your goal in a very short amount of time, on average only about 30 steps. This is the power of a stepwise process where each successful invention is preserved and built upon. The RNA world hypothesis serves as a mathematical bridge in this context, collapsing the separate probabilistic hurdles of information storage and enzymatic catalysis into a single, more likely molecular event. Once a single self-replicating molecule or a small catalytic cycle emerges, the rules of the game change from pure chance to a form of molecular evolution. Any variation that improves the speed or accuracy of replication will naturally become more common. This selection process allows complexity to ratchet up over time.
This leads to the question of search spaces. The chemical space, the total number of possible ways to arrange atoms into molecules, is vastly larger than the physical space of the universe. Exploring this space to find the specific needle in the haystack that is a living cell seems impossible. Yet the haystack might be filled with many more needles than we realize. We often assume life must use our specific 20 amino acids and 4 nucleotides, but nature offers a vast palette of up to 100 amino acids and at least a dozen nitrogenous bases. If there are millions of different ways to build a functional self-replicator, then the probability of finding at least one of them increases significantly. Furthermore, proteins are not fixed, rigid sequences. Studies in functional equivalence show that some molecules can have 30 to 50% of their amino acids differ and still perform the same biological task. This suggests that the target for life is not a single infinitesimal point, but a much larger and more accessible region of chemical space. If many different sequences can achieve the same function, the statistical mountain of abiogenesis becomes much easier to climb. The timing of life's emergence on Earth adds another layer to this mathematical debate. While the last common ancestor likely dates to 4.0 billion years ago, the earliest chemical signatures of biological activity appear in the geological record as far back as 4.2 billion years ago, placing the mathematical window for abiogenesis in a surprisingly narrow slice of time, just a few 100 million years after the Earth formed. To many, this suggests that abiogenesis must be a relatively easy and probable event.
However, a more cautious approach using Bayesian statistics suggests that we cannot draw such a firm conclusion from a single data point. This analysis, notably explored by Spiegel and Turner, examines how the discovery of our own existence affects our estimate of the probability of life. We are sentient observers who require billions of years of evolution to exist. Because the Earth has a limited lifespan, eventually the Sun will expand and make the planet uninhabitable. Life had to start early for there to be enough time for us to evolve and ask the question of how we began. This is a form of selection bias. The technical heart of this "is life rare" debate lies in the choice of a Bayesian prior. Spiegel and Turner's analysis highlights the difference between a log-uniform prior, which assumes life could be equally likely at any scale of probability, and an uninformative prior. Their work suggests that the fact that life started quickly on Earth is consistent with life being either very common or incredibly rare in the universe. Without a second independent instance of life, the math remains frustratingly inconclusive. The mathematical challenge is further complicated by the halting problem of biological information and the concept of biosemiosis. Some researchers, such as David Abel, argue that life is not just a matter of physics and chemistry, but a formal system of computation. In this view, the laws of motion and chemical attraction do not perceive, value, or pursue usefulness or functionality. A prebiotic environment has no reason to select a molecule because it is a good catalyst. It only favors molecules that are chemically stable. Yet, every process in a living cell is highly functional and goal-oriented. This functional gap is illustrated by the sheer efficiency of biological catalysts. For instance, the enzyme phosphatase accelerates vital cell signaling reactions that would otherwise take 1 trillion years to occur through undirected chemistry. Without the purposeful shortcuts provided by such enzymes, the chemical processes of life would essentially grind to a halt, far outlasting the stars themselves. Bridging the gap between the aimless reactions of the inanimate world and the purposeful orchestration of a metabolism requires a mechanism that natural science has yet to fully define. This is the halting problem of biosemiosis. The laws of physics can describe the motion of atoms, but they cannot explain the emergence of a code that assigns meaning or function to those atoms. To reconcile these views, some scientists look to the sheer scale of the natural laboratory that was the early Earth. While a human researcher might spend 70 years trying to recreate abiogenesis in a few glass beakers, the early Earth had hundreds of millions of years and an entire global ocean. The number of permutations and combinations that could be tested in such an environment is almost beyond calculation. If we think of the right chemical combination as a specific shuffle of a deck of cards, we know that every time we shuffle a deck, we likely create a sequence that has never existed before. A single shuffle of a standard deck results in one of roughly 8 times 10 to the power of 67 possible combinations, a sequence so unique it has likely never been repeated in human history. With enough shuffles occurring simultaneously across the planet, even highly improbable events become statistically certain. The physical settings discussed in previous chapters provide the necessary concentration math to make these shuffles more effective. Recent experiments involving sapphire substrates and mineral surfaces suggest that the microscopic chambers of the early Earth could have increased the concentration of building blocks by 3 orders of magnitude. By confining reactions to a small space, the effective concentration of molecules is increased by thousands of times, making the formation of complex polymers much more likely than in an open, dilute environment. This concentration effectively stacks the deck, increasing the frequency of molecular collisions and the likelihood of a successful assembly. Recent mathematical frameworks using information theory and algorithmic complexity are now being used to quantify exactly what it would take for a protocell to spontaneously assemble. These studies suggest that while chance plays a role, there may be undiscovered physical principles that govern how systems of matter become more organized and functional. The search for these principles is one of the grand challenges of biological physics. If such laws exist, they would mean that life is not a fluke, but a predictable outcome of energy flowing through matter under the right conditions. The debate remains an impasse because we lack a second example of life. While scientists have successfully created synthetic life in the laboratory, engineering bacteria with entirely man-made genomes, this is fundamentally distinct from recreating a natural abiogenesis event. We can assemble the hardware of life from a blueprint, but we have yet to witness the blueprint writing itself from the chaos of a prebiotic soup. If we were to find even a single microbe on Mars or Europa that resulted from an independent abiogenesis event, the mathematical argument for inevitability would win instantly. It would prove that the interstellar pantry of the solar system is capable of producing life twice in the same neighborhood, making it a near certainty that the universe is teeming with biological activity. Until then, we are left to weigh the astronomical odds of molecular assembly against a massive scale of geological time and the potential for stepwise cumulative complexity. The mathematical scrutiny of our origins forces us to acknowledge just how much we still do not know about the transition from the inanimate to the animate. Whether life is a common feature of the cosmos or a singular miracle, the fact that a system of atoms can eventually learn to calculate its own probability is a profound reality. If the laws of physics are indeed blind to usefulness, how did the first chemical sequences begin to read their environment and respond with purpose?
The story of our origins suggests that while life's ingredients are universal, its successful establishment was a transformative, winner-take-all event. From the singular genetic code of Luca to the closed door created by an oxidizing atmosphere and biological competition, our lineage didn't just survive, it reshaped the Earth to ensure its own monopoly. Whether this was a statistical miracle or a chemical inevitability remains the ultimate question. If life is a robust consequence of planetary physics, then weird life may still hide in a shadow biosphere or across the stars. We are the descendants of a 4-billion-year-old success story that effectively ended the era of spontaneous creation on our world. We are a single branch of a tree that might have been a forest, standing as a testament to a journey from stardust to sentience. Thank you for listening.